Hello and welcome everybody to my channel ominibioentrance, and today we will be discussing CSIR PYQs of the year 2014. If you are new to my channel, please do like, share and subscribe. Coming to the year June 2014, membrane bound and free ribosomes are structurally identical but differ only at a given time in terms of association with acylated proteins. These are part B questions. So answer here is option 1, acetylated proteins. Then next one. Histone acetylases and chromatin remodeling complexes are recruited to specific regions of the chromatin by gene activator proteins. Answer is option 1. Next question. Each origin of replication is activated only once. This is achieved because pre-replicative complex can only form in the G1 and replication can only be initiated when pre-initiation complex is disassembled at the beginning of the S phase. So that is why each origin of replication is activated only once. Answer is option 1. The next one. The homologous genetic recombination is a DNA repair process referred to as recombination repair. Which one of the following statement is incorrect for the recombination repair? DNA polymerase 3 stalls at the site of damage. That is correct. DNA polymerase 3 leaves a gap in the daughter strand. That is also correct. The gap is filled by recombination between complementary parent strand homologous to daughter strand and the gapped daughter strand. That is also correct. Homologous recombination process is catalyzed by topoisomerase 2. Topoisomerase is not involved in recombination repair. So option 4 is incorrect. Then next one. During protein synthesis in prokaryotes, the peptidyl transferase activity required for peptide bond formation is due to. It is due to ribosomal protein L26. Answer is option 1. The next one. After activation of the promoter by the DNA binding activity of a transcription factor, a co-activator is recruited at the region targeted for transcription, which in turn creates a binding site for a chromatin remodeling complex. Which one of the following activities of the co-activator is responsible for the recruitment of chromatin remodeling complex? It is histone acetyl transferase activity. Answer is option 3. Then next one. Prior to transcription, chromatin changes from an inactive state to an active state by various factors in a stepwise manner. Which one of the following is involved in the initial step during activation of a chromatin loop? So during activation of a chromatin loop, it is topoisomerase 1. Answer is option 4. The next one. Virus-induced gene silencing is a process that takes advantage of the RNAi-mediated antiviral defense mechanism. Which one of the following ultimately guides siRNA to degrade the target transcript, that is mRNA? It is single-stranded RNA. Answer is option 2. Now coming to part C questions. Amino-acyl tRNA synthetases are very specific for amino-acylation of tRNAs with the correct cognate amino acids. However, there is a possibility of a mismatch between the tRNA and its cognate amino acid. This error is corrected by inherent proofreading activity of the amino-acyl tRNA synthetase. In case of two very similar amino acids, namely valine and isoleucine, isoleucyl tRNA synthetase employs the following possible approaches for an error-free amino-acylation. So it removes an incorrect amino acid by hydrolyzing the amino-acyl AMP linkage following the first reaction step. So this A is correct. It is activated for proofreading activity leading to breakage of the bond between the wrong amino acid and tRNA. That is also correct. It has an intrinsic ability to recognize the structural difference between amino acids, leading to abortive elimination of the non-cognate amino acid. That is not correct. It gets sequestered in the second step with the wrong amino acid, and that freezes the amino acylation process. That is also not correct. So the option here is option 1, that is A and B. The next one. Attenuation is a mechanism involved in the regulation of tryptophan operon in E. coli. When tryptophan levels are high in the cell, region 2 of the TRPL is blocked from pairing with region 3. This allows the pairing of region 3 and 4, leading to the formation of row-independent terminator. What would be the structure of the TRPL region in E. coli cells where protein synthesis has been inhibited? So, region 1 and 2 will pair, allowing 3 and 4 to pair, leading to attenuation. Answer is option 2. Then next one. MicroRNAs have recently been shown to play significant role in the fine-tuning of gene expression. Some miRNAs induce gene silencing by binding to mRNAs and inducing inhibition of translation. On the other hand, there are miRNAs that bind to mRNAs and activate their degradation. The following characteristic can be applicable to the miRNAs that inhibit mRNA translation. miRNA is partially complementary to the region of the target mRNA in the 3' UTR. That is correct. miRNA always base pairs with mRNA around a AU-rich sequence. That is not correct. miRNA base pairs with mRNA through 6 to 7 nucleotide at its 5' end, referred to as seed sequence, as well as a few additional bases elsewhere. That is correct. So A and C are correct. Then next one. The following statements are made on DNA replication. Which of the following is the correct combination? Replication fork is a branch point in a replication eye or bubble. That is correct. A replication bubble contains 2 replication forks. That is also correct. See, if you see, DNA replication is continuous according to the interpretation made by Okazaki. Okazaki told it is discontinuous, not continuous. Multiple priming events are required for both leading and lagging strand to initiate DNA synthesis. Leading strand requires only one primer, whereas lagging strand requires multiple primers. So that is also not correct. So the answer here is option 1, A and B. Then next one. In order to identify the regulatory region of a novel promoter sequence shown above, 4 150 base pair deletion constructs were made in a luciferase reporter system as indicated above in the boxes A to D. After transfection, the observed levels of promoter activity percentage as analyzed by luciferase assay of all the construct is indicated in the right of the figure. Identify the best correct combination of regions in the options given below that indicate the presence of a positive and a negative regulatory element. So from this deletion construct, you have to identify which for the presence of a positive and a negative regulatory element. Now, if without any deletion, it is showing 100% activity. Now, if there is a deletion in region A, the activity goes down drastically, that is 20%. That means A was a positive regulatory element. That is, it enhanced the activity of the promoter. Now, when A was deleted, it drastically fell down to 20%. So, A must be a positive regulatory element. Now, C is having 140%. That is, it has increased the activity more than 100%. That means C must be a negative regulatory element. So, the answer here is option 2. Coming to the year December 2014. Which one of the following statements about eukaryotic translation is not true? In eukaryotic translation, ribosome binding site on mRNA is called Cossack sequence. Initiator tRNA is tRNA FMET I. That is not the initiator tRNA in eukaryotic translation. So that is the one which is not true. Then next one. During replication, the RNA primer is degraded by the 5' 3' exonuclease activity of flap endonuclease 1. This happens in eukaryotes. Answer is option 2. Then, small nucleolar RNAs used to process and chemically modify RNAs are called SNO-RNAs. Answer is option 3. Then next one. Leader sequence in some of the protozoan parasite is transcribed elsewhere in the parasite genome and gets joined with several transcripts to make the functional RNA. The joining of the two transcripts occur by the process of in protozoan parasite, it is by trans-splicing. Answer is option 2. The next one. A gene producing red pigment was placed near centromeres of a fission yeast and thus subjected to the position effect variegation and produce white colonies. Naturally, it will produce white colonies because centromere is constitutive heterochromatin region. So, translation or transcription will not take place there. So, there will be no expression of the red pigment. Okay. Now, next statement. A screen for mutants that increase the red pigment production was undertaken. Sorry. Which of the following genes when mutated is likely to produce this genotype? So it is histone deacetylase. Now histone deacetylase is responsible for heterochromatin formation. So when mutated, the gene will be expressed as it gets converted to euchromatin and thereby giving rise to red pigment. Then next, a promoter deletion study was done in order to determine the binding sites for the transcription factor on the promoter, which is activated on treatment with drug X. The following constructs were made. Luciferase assay revealed the following results. The following statements can be made. Region between 1800 and 1210 contains a binding site for the activator. So when treated with the drug, luciferase activity has increased, whereas in the control it remains the same in all the cases. As well as 1210 also the activity got increased upon treatment with the drug. So it is the site for it is the binding site for the activator. So A is correct. Now region between 868 and 1210 contains a binding site for repressor. That cannot be true. It is because 1210 till 1210 it is the binding site for the activator. Then region between -868 and 432 contains a binding site for the repressor. That is why the activity was reduced drastically upon treatment with drug. So C is also correct. So your answer here is option A and C. That is option 1. Then next one. Total RNA was isolated separately from cytosol and nuclei of human cells growing in a cell culture. Each sample was mixed with a purified denatured fragment of a DNA corresponding to a large intron of a housekeeping gene and incubated under renaturation conditions. Given below are the statements made about the outcome of the experiment. So, RNA isolated from the nuclei will form DNA-RNA complex duplexes because of the presence of introns in the primary RNA. That is correct because RNA isolated from nuclei is pre-mRNA and it will have introns. So, DNA-RNA hybrid formation could be possible. Then next one, cytosolic RNAs usually will not form RNA-DNA duplexes. They too form RNA-DNA duplex. If you see the statement D, cytosolic RNA will form RNA-DNA duplexes because unspliced cytosolic RNAs are exceptionally stable. So A and D is the correct answer. Then next one. Following are certain statements related to eukaryotic DNA replication. Which of the following combination of above statement is correct? The genome of multicellular animals contain many potential origin of replication. That is correct. During early development, when embryos are undergoing rapid cell division, origin sites are uniformly activated. That is also correct. Pulse chase technique is used to label sites of DNA replication. That is also correct. So A, B, C are correct, whereas D is incorrect. Then next one. Puromycin is an antibiotic used to inhibit protein synthesis. Given below are few statements about the antibiotic. Now before coming to the statements, antibiotic puromycin target both prokaryotic and eukaryotic cells. Then molecular targets include peptidyl transferase center of large ribosomal subunit. Then what is the consequence? It results in chain terminator. It mimics the 3' end of amino acyl tRNA in A site and act as acceptor for nascent polypeptide chain. Now read the statements. It enters the A site of the ribosome, E site of the ribosome, where it prevents the release of deacetylated tRNA after the action of peptidyl transferase. That is not correct. It enters the A site. The next one. It blocks the translocation process by binding to the translocation factor EFG. It doesn't bind to any translocation factor EFG. Puromycin resembles the initiator tRNA fMetno and binds exclusively to P site. It binds to A site and not to P site, and it doesn't resemble tRNA fMet. Then it resembles the amino acyl tRNA and bind to the A site of the ribosome. That is correct. Puromycin inhibits only prokaryotic protein synthesis. It inhibits both prokaryotic and eukaryotic protein synthesis. So F is correct. So D and F are the answer. So answer here is option 3.